Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the chemotaxis in bacteria. This topic is related to cell signaling process, and here I will try to explain this very briefly. And you can obviously consult any book that is related to cell biology just after seeing this video. And I have uploaded many videos related to cell signaling process and cell biology obviously, so you just check out all those videos. And I it's my humble request to the newcomers that please you share and subscribe this channel if this channel is helpful to you. You just give your support to me for increasing the growth of this channel. Okay, so let's start. That is the chemotaxis in bacteria. So here the chemotaxis meaning is that movement of organisms in specific direction in response to chemical stimulus. So what is called the chemotaxis? Chemotaxis is actually the movement of organisms. Generally bacterial movement is is told here, that the movement of the bacteria, you can say, towards the specific directions, okay, and in response to the chemical stimulus. This is called the chemotaxis. Okay, so bacterial flagellar motion is rotatory in nature. So generally you all know about this. This is the bacteria. Sorry, this is the bacteria and this is the flagella. Okay, so the flagellar motion is seen, it is rotatory in nature. Okay, and in there that means the membrane of the bacteria that contains the proteins like that MOT A proteins and MOT B proteins. Okay, and another is FLIG. So MOT A and MOT B and FLIG all together create a proton channel. Okay, create a proton channel, and that drives the relation of the flagella. Okay. Okay. I stopped here just for checking this word. This is not the relation, this is the rotation. Okay. So what is saying that this MOTA and MOTB and FLIG all create a proton channel, and that helps in the rotational movement of the flagellum. And the rotation that may be clockwise or counterclockwise. Two types of rotations are seen, clockwise or counterclockwise. So here the flagella, when rotates clockwise, that forward motion ceases and the cells tumble. Tumble means fall down. Okay. So in case of clockwise motion of the flagella, that showing the forward motion, but it stops after some times and the cells tumble. But in case of counterclockwise rotation of the flagella, that creates the impulse or that creates the forward motion or run. So which one is very important for the bacterial motion? That is the counterclockwise rotation. Okay, so counterclockwise rotation that impart the forward motion, but the clockwise rotation that seizes the forward motion and the cells tumble here. And in absence of the chemical gradient, obviously chemotaxis we will learn. So here the role of the chemicals is present always. Okay, so if chemical gradient is absent, okay, so the movement is seen in a random fashion. Okay, so if chemical gradient is absent, so the random fashion movement is seen. But chemical gradient if present, then random movements become biased, or you can say that the movement is seen in a unidirection way. Okay, So as the bacterium senses that it is moving towards the higher concentrations of the attractant, here two words are very important. One is the attractant and another is the repellent. Okay, if the attractant present or bacteria senses the presence of attractant, so the runs become longer. Clear? The runs become longer when the bacteria senses the presence of attractant. But when they senses the presence of repellent, so in case of repellent, it's saying that the same general mechanism applies, but it is decrease in concentration of repellent promotes run. That means if repellent concentration is less, that helps in that promotes in the run. Okay, or you can say that promotes the forward motion. But repellent present in high concentration, so there is the amount of the forward motion or forward movement that becomes very slow. Clear? So chemotaxis is mediated by the transmembrane receptors and the phosphorylation relay system. Two processes are very important. One is the phosphorylation system, relay system, and the presence of the membrane receptors. Both help in the chemotaxis process of bacteria. And here it depends on two component signaling pathway. Two component signaling pathway, very important, that are activated by the histidine kinase. Okay, that activated by the histidine kinase with receptors or histidine kinase associated receptors. The chemotaxis receptors are methylated during adaptations and so are called the methyl accepting chemotaxis proteins. So in sometimes or when they show the adaptations, in that situations the chemotaxis receptors that becomes methylated, and in that situations they are called the methyl accepting chemotaxis proteins, MCPS. Okay, and the phosphorylation relay system that enables the chemotaxis receptors to control the flagellar motor. Okay, so here the phosphorylation system, relay system, that also controls the flagellar rotation. Okay. So how this phosphorylation relay system helps in the flagellar rotation, so we will learn in after this because the space is very short. So binding of repellent that increases the activity of the receptor. So when the repellent can bind with the receptor, then the activity of the receptor that increases. And which binds CHEW. CHEW is one kind of adapter protein, and also with CHEA, that is the sensor proteins, and it acts as the histidine kinase. Okay, and when they bind with the CHEW and CHEA, thereby stimulating CHEA to phosphorylate itself on histidine. Okay. So what is happening here? That means just repellent bind and the activity of the receptor increases. So at that time they bind with the CHEW and CHEA. So when CHEA that becomes activated, they try to phosphorylate itself on histidine. Okay. So in that way the forward motion, or you can say the rotatory motion, or the chemotaxis process, this starts. Okay, so we will learn about this in the next part. Next part means after rubbing this portion, I will try to explain this one. So here the CHEA that quickly transfers its covalently bound high energy phosphate directly to aspartate of CHEY. Okay, so After phosphorylation, what happens when CHEA, just after phosphorylated, it transfers the phosphate directly to the aspartate of CHEY, and CHEY, this is the receptor regulator protein. Then to generate CHEY phosphate, so after transferring, it forms CHEY phosphate, and the phosphorylated CHEY then dissociate from the receptor and diffuses through the cytosol and binds to the flagellar motor protein and causes it to rotate clockwise. So what is happening here? When CHEY that becomes phosphorylated, it dissociates from the receptor and diffuses within the cytosol, okay, and then binds with the flagellar protein, that is the motor, and induce to rotate the flagellum in clockwise direction. So what is happening? So resulting the tumbling. So not the forward motion is completed, so cells become tumbles. Okay, and phosphorylated state of the CHEY that remains only for few seconds. And the protein CHEZ, this is another protein, this accelerates the dephosphorylation of CHEY. So one protein, that is CHEA, that helps to phosphorylate CHEY, and that creates the clockwise motion or clockwise rotation. Clear? But another protein, that is CHEZ, that dephosphorylate the CHEY, and as a result, inactivations of the CHEY it is seen. And so binding of an attachment has the opposite effect. So its opposite effect is that when inactivation of the CHEY is occurring, that creates the counterclockwise rotation. Okay, flagellar counterclockwise rotation. And here it inactivates the receptor and therefore the decreases phosphorylation of the CHEA and CHEY, which result the counterclockwise flagellar rotation. So just when the dephosphorylation of the CHEY is occurring, that helps in the counterclockwise rotation of the flagella, and it's due to this the forward motion is seen or forward movement is seen. Each of the phosphorylated intermediates, so it's that phosphorylation or dephosphorylation that present within the cell, not cell, present within the it stay within the bacteria just for 10 seconds and enabling the bacterium to respond very quickly with the environment. Okay, so this phosphorylation and dephosphorylation, that is the continuing process and it occurring within few seconds. So that's why bacteria can easily respond to the environmental changes. And the response to an increased concentration of attractant or repellent is only transient. So here this process is occurring, but here another role of the concentration of the attractant and repellent is seen, but this is only transient. That means even if the higher level of ligand is maintained, as the bacteria desensitize or adapt to the increased stimulus. Clear? What is saying that? That means here the ligand level or the level of the ligand concentration is higher, but bacteria desensitize or adapt to the increased stimulus. So if the process is not continuing, so if chemical present or not present, or bacteria sensitize or desensitize, that will not be affected. Okay, so this process must be continuing, and then you can say this is the concentration of the attractant or repellent that affect on the bacteria's rotational movement. Clear? And here another important thing is that adaptation also is mediated by the process of methylation, and here important catalyst is that methyl transferase, CHER. And here the name of the proteins that already we have learned in this topic, like that CHEA, EW, CHEY. So these are written here just for as a gist or as a summary, so you can easily understand about the proteins or the short informations related to the proteins. Okay, so this is the actually chemotaxis process in bacteria that is seen in the cell signaling process, or this occurring due to the cell signaling process. Clear? So I have to draw a rough structure or rough diagram. Just after rubbing this, I will try to draw. So this is the rough structure. This is the bacteria. Okay, this is bacteria, and this is the flagellum of the bacteria, and clockwise rotation is seen here. So what is happening? Repellent first bind with the chemotaxis receptor, then this is the CHEW and it becomes activated, then CHEA that is activated and phosphorylated, then it try to activate CHEY, and after activation the phosphorus that dissociated and CHEY is formed here. So here the clockwise rotation is seen. So this is the very rough structure of the clockwise rotation, and counterclockwise rotation I have not drawn here. Okay, only I want to I try to explain about the clockwise rotation by this brief diagram or brief sketch. So this is the chemotaxis in bacteria. So I think you can totally understand about this. So in the next video I will try to another topic related to the cell signaling process. So please you continue with my channel, and you will try to see the another many topics related to the cell signaling process that are all uploaded in my channel. So please you see about this topic. Okay, thank you.